Before we start, we need to introduce you to GOLPH3, less conveniently called Golgi phosphoprotein 3. It is a transmembrane protein which is required in part for the structure and function of the Golgi. The gene encoding it is found in human chromosome band 5p13, which is commonly amplified, showing an increase in copy number in various human solid tumours. In prostate cancer, it is located in the cytoplasm of tumour cells, and the mRNA level and protein levels of GOLPH3 have found to be overexpressed, increasing with tumour progression. Due to this, it has been marked as a metastasis-associated gene and an oncoprotein candidate. In 2017, over 3,000 Australian men are expected to lose their lives to prostate cancer. The lethality of prostate cancer is not associated with the size of the tumour, but rather the metastatic ability of these cells to other organs. Cancer metastasis is the success of cancer cells to detach from the primary tumour, invading through the extracellular matrix and thriving in distant organs. While treatments exist for localized prostate tumours, there is no cure for metastatic prostate cancers. In the normal prostate epithelium, GOLPH3 is expressed and interacts with the proto-oncogene tyrosine protein kinase SRC. It removes and replaces tyrosine 416, normally present in the SRC binding pocket, and this interaction causes a phosphorylation of SRC. In the phosphorylated state, SRC is active. Activated SRC then goes on to phosphorylate the tyrosine on P65, a subunit of nuclear factor Kb. This promotes P65, which binds to the promoter of matrix metalloproteinase 9, MMP9, to initiate its transcription, increasing levels of MMP9. MMP9 degrades the basement membrane and extracellular matrix, important in tissue remodeling and repair. In prostate cancer, GOLPH3 is overexpressed due to the amplification of the chromosome band 5p13. This results in the upregulation of SRC and consequently of P65. Elevated P65 increases the transcription of MMP9 to the point of extensive breakdown in the basement membrane and extracellular matrix. This is responsible for the epithelial to mesenchymal transition of the prostatic epithelium, in which they lose their cell adhesion and polarity and gain mesenchymal migratory properties. The epithelial to mesenchymal transition enables the primary tumour to undergo intravasation, that is, to enter the circulation and metastasize to a distant site, usually the bones in the case of prostate cancer.
To limit the effect of GOLPH3 in prostate cancer, GOLPH3 expression is silenced by DNA-directed RNA interference through lentiviral gene therapy. First, a plasmid is constructed that encodes for short hairpin RNA molecules, which silence the expression of GOLPH3. This plasmid is then inserted into the host cell by a lentiviral factor, which introduces the plasmid directly to the targeted prostate cancer cell by antibody-antigen interactions. The membranes of the host cell and the lentivirus then fuse, causing release of the plasmid into the cytoplasm, which then migrates into the nucleus. From there, the plasmid interacts with RNA polymerase II, causing expression of the GOLPH3 specific shRNAs. These shRNAs exit the nucleus and form a silencing complex, which cleaves and replaces the mRNA encoding for GOLPH3, hence silencing the gene. Effectively, ddRNA interference aims to activate the endogenous RNA interference pathways, rather than inducing interference synthetically. The advantage of introducing a plasmid to the host cell, rather than directly introducing synthetic shRNA, is that it bypasses the intrinsic immune response of the cell. Toll-like receptor 3 is a protein expressed across the membrane of the cell, which triggers an innate immune response once a pathogen is recognized. When shRNA is introduced directly to the host cell, like what is done in a traditional RNA interference, it activates these receptor proteins. This immune response can be very severe and has caused death in human trials. ddRNA interference, however, bypasses this issue by producing shRNA from within the nucleus of the host cell, the outcome being that there is no immune response because there is no TLR3 shRNA interaction. Additionally, this treatment is also very cost effective, considering that a very low dose of 5 plasmids are required per patient. The main area of concern is that there is a potential of toxicity to the patient if shRNA is overexpressed, so it is imperative that this is closely monitored during trials.